All right, welcome back, Wayne County Community students. We're going to continue on with the physiology portion of anatomy and physiology, where we're going to explain how blood transports gases. Of course, those gases are oxygen and carbon dioxide. So how does blood transport these gases? Let's take a look at that. One way is as a solute, where the gas is in a fluid. So this is how blood transports gases. Okay? Okay. One, it has a solute. And as a solute, the gas, oxygen, carbon dioxide, once they're in the blood, they immediately dissolve in the plasma. Because fluids can only hold really small amounts of gas in solution. So this probably, taking oxygen and carbon dioxide as a solute, probably isn't the most optimal way to transport a gas. This is Henry's law that says fluids don't really hold gases too much, so once the gas gets in a plasma, into our plasma, it really doesn't, it dissolves and it's really not carried that far. Another way though is to transport gases this way. In a chemical combination or chemically combined. Okay? A chemical combination, this is where you see like the majority of the gases, the majority of the oxygen and the carbon dioxide. They form a chemical union, right, with other molecules. Just say, for example, hemoglobin. And then, of course, you know hemoglobin is a plasma protein. And then this gas is going to be bound to another molecule, in this case hemoglobin, and that will decrease the plasma concentration. And now more gases can diffuse into the plasma. And this way, large volumes of gases can be transported this way. When oxygen is bound to hemoglobin, and remember hemoglobin is HbO2, we call that an oxyhemoglobin. The majority of the oxygen, as you learned before when we did the heart, this is the way our oxygen, the majority of it, is transported, attached to hemoglobin, right? You remember it on a red blood cell about the iron and the heme group and the hemoglobin and the four polymers of hemoglobin that make up one hemoglobin. Well, oxygen is attached to that hemoglobin, and that is the major route of oxygen transportation. So if I ask on the exam, what is the major route of oxygen being transported? Please remember, for oxygen, it's an oxyhemoglobin. Oxygen is transported attached to hemoglobin, okay? Oxygen associates with hemoglobin so rapidly that about 97% of hemoglobin has bound with oxygen by the time the blood leaves the lungs capillaries to return to the heart. So that's a pretty good affinity for oxygen and hemoglobin. And that's really what you want for sure as aerobic beings, right? Now that we know how oxygen is transported via the hemoglobin, let's take a look and see how the carbon dioxide is transported. So the transport of carbon dioxide. Now the carbon dioxide is a little more detailed And there are going to be 3 ways that carbon dioxide is transported. 3. All right. So we say carbon dioxide is transported from the tissues to the lungs in 3 forms, right? It's going to be coming from the tissues to the lungs, so then we can breathe that out as waste. The carbon dioxide will be breathed out as waste. One way is dissolved. Now, we just talked about gases being a solute or being dissolved, right? This is carbon dioxide dissolved in plasma. We just learned when we were talking about gases gases in general being transported as a solute, in a liquid. Well, you know that carbon dioxide will dissolve in plasma and transport it as a solute. We just discussed this. So would you think that carbon dioxide being dissolved in plasma is a great way to transport gases or to transport this carbon dioxide? The answer to that would be it's okay, but it's really only 10% of carbon dioxide can be carried in the blood as it's dissolved in plasma. So this is an okay method, but not great, because you know carbon dioxide is our waste product, so we want to get rid of a whole lot more than just 10%. Let's look at it in this way. As a carbamino compound. This is where carbon dioxide is bound with an amine group. So here, let's dissolve. Here, carbon dioxide is bound with an amine group. And you know an amine is an NH2, for example. And that's going to be known as a carbamino compound. Carbamino compound. Hemoglobin is still the main protein to combine with carbon dioxide, and this is known as carbamino hemoglobin, right? And it looks something like this. So carbamino compound, when carbon dioxide is bound with an amine group, and this will be the amine group NH2, this is known as carbamino compounds. Hemoglobin, if you recall from our heart lecture, hemoglobin, the hemoglobin molecule has the globulin parts to it, 4 of those per hemoglobin molecule. The carbon dioxide attaches to that globulin part. You remember that? Great. So the hemoglobin is still the main protein to combine with carbon dioxide. Remember, the carbon dioxide binds to the globulin part of the hemoglobin, or the globulin part. And now, with the carbon dioxide being bound with an amine group as a carbamino, hemoglobin is still important to this carbon dioxide. That carbamino will then combine with the hemoglobin, and the carbon dioxide becomes a carbamino hemoglobin. This is the carbon dioxide part, the amine group, which is here, and then the hemoglobin, which is there, right? It looks something like this, hemoglobin, and then the amine group. Here is the carbon dioxide, and this is just the H that would have gone on with the amine group here, the N. So that's a carbamino hemoglobin. Here's the hemoglobin, the NH is the amine, and the CO2, COO, is the carbon group, carbamino. This only carries, here again, it's double what the dissolved group would carry, but it carries just 20%. We need a lot more carbon dioxide out of our system than that. So I'm just going to highlight these so you will remember that dissolved and carbon dioxide dissolved in plasma is not that great a way to transport a gas, or it being combined to an amine group and a hemoglobin. It's okay, but it's not optimal. Let's look and see the third route of way to transport carbon dioxide. So I'll put it here. This is the third way, right? Third way to transport carbon dioxide is as bicarb. Oops. We call it bicarb or bicarbonate. This is the third method. Bicarb is just short for bicarbonate. So I always usually just say bicarb. Carbon dioxide is also carried in the form of bicarbonate ions. So here, I have to steady this board so I can write on it. Carbon dioxide Carbon dioxide is also carried in the form of bicarbonate ions, right? Let's see how that actually pans out. How does that look? So let's draw, we're going to have the eye kind of look at a red blood cell, kind of sideways. So let's just draw a red blood cell, okay? You know, this is the biconcavity here, here, and here. So you have carbon dioxide come in to the red blood cell, carbon dioxide. It's going to combine with water. Then it's going to use a really great enzyme that we introduced when we did the heart lecture. See, I want you to notice I keep saying that, referring back to the heart lecture, because everything is congruent. Everything combines. You can't just learn one thing and not expect it to trickle over to something else. So I just want you to know that everything is cumulative, right? When you learn about science, you have to remember some other stuff from before, previous lectures sometimes. So carbonic anhydrase is the enzyme here, and that will form carbonic acid. right, so I'm gonna write the word carbonic anhydrase here so you can see it better. But I want you to know that's the enzyme that's used here. So carbon dioxide, is part of our waste. So it comes into the red blood cell, and we already learned how we breathe it out by a pressure gradient difference. But then it does combine with water, uses this enzyme that's on practically every one of our cells. Carbonic anhydrase is very important to the cells. And it forms this carbonic acid. Carbonic acid then dissociates to a hydrogen ion and bicarb, right? To bicarb. Right here. You can see where you have the COO3, COO here, that right here is the CO2. So that is the carbon dioxide that's hidden in the bicarb, all right? Now another part about this is that the hydrogen that's left over combines with the hemoglobin, and it dissociates and becomes a hydrogen ion plus hemoglobin. I'm not so much concerned with that. I mainly want you to kind of get this concept with the carbonic anhydrase, and the main concept is that bicarb, 70% of our carbon dioxide is carried this way as a bicarb ion. That's the main gist of this story. So we see 10% here is dissolved, not a great way to carry carbon dioxide. 20% is a carbaminohemoglobin molecule, still not that great way of carrying carbon dioxide. But here, as a bicarbonate, carbon dioxide can be carried quite efficiently, by the way, and 70% of our carbon dioxide is in the form of bicarb. That is the main gist. And on this red blood cell, I'm just showing how that occurs. Now, one other thing that's pretty important about this is there's this, so let me move this 70%. Let me put it somewhere else because I got to draw something else here. Where can we put this? 70%. All right. So what I want you to know is here, I'm just going to draw a little circle there because I need to take this bicarb out so you can get rid of the carbon dioxide. But what has to come in to keep this equation balanced? Here's a plus, here's a minus that neutralizes. When this minus, this carbon dioxide goes out to the plasma, this chloride that's in the plasma, Cl, must come in to keep this cell balanced with another, let me show you that, that's Cl, chloride, all right? That negative comes in to replace the negative that's leaving, that ion that's leaving, and it makes this red blood cell stay balanced. When that shift occurs, that is called a chloride shift. Chloride shift. Is the chloride coming in to replace this negative ion that left, the bicarb. Please remember, 70% of our carbon dioxide leaves through a bicarbonate. Now what I'm going to do is write all of this up here to explain what's going on with this red blood cell. I'll write it, and that will be actually the last slide on this this tape. It'll just explain this red blood cell and what's going on in words other than just a drawing. It gives a pretty good depiction in the text, but I'd rather you have something from my notes as well. I'll put that on later. But right now I'm going to finish up and talk about Yeah, I'll just explain that later. I'll read it, and then that way I'll write it at the same time later. Some of the carbon dioxide that's entering the red blood cell is going to combine with water. So some of the carbon dioxide that's entering the red blood cell will combine with water to form carbonic acid. That's this. Using an enzyme, carbonic anhydrase, here, right? That's present inside each cell. Carbonic acid then dissociates to form hydrogen ion and bicarb. Hydrogen ion and bicarb. The hydrogen ion combines with hemoglobin while the bicarb diffuses down its concentration gradient. So it's going to go from a higher concentration to a lower concentration into the plasma. As the bicarb leaves each red blood cell, chloride enters and prevents an imbalance in the negative charge. This is known as a chloride shift, where you get a counter transport of negative ions. And that helps keep this red blood cell balanced. I'll put that in words on the board so you can understand that. Read that anyway for your pleasure. And that will be the last slide that will be on. So this allows a greater carrying capacity of carbon dioxide in the blood. One thing I want to, another little chart I want to put on, and then I'll write the other stuff up, and then we'll be done with this lecture. And then this will be all that will be required for the lymphatic and respiratory exam, right? Okay, let me just go over quickly about the oxygen dissociation curve, just to show you something. So you have this, well, this is moving, I don't know. Am I still in frame?
Yeah.
All right, so let's see. Very quickly, I just want to introduce something known as the Bohr effect, because I think I'm going to ask something about that. I'll have to look and see what these exams have, but just so that you will have seen this curve. So we consider this one normal. Well, let's see, this one's normal. Okay. And this one is going to show what? A shift to the right. See how from normal you get a shift to the right? Well, a right shift, always label what your graph is. It's an oxygen dissociation curve, right? Really, I should have my X and Y axis labeled too. Always do that, but I'm not doing that today. This shift from normal, or how where you have your oxygen and hemoglobin, this oxygen dissociation curve, this shift to the right is an indication of how hemoglobin affinity. So a right shift in the oxygen hemoglobin dissociation curve is due to a partial pressure of carbon dioxide. It's also known as the Bohr effect. When the curve shifts to the right, there's a decrease in affinity to oxygen, right? This facilitates the unloading of oxygen to the tissue. So it helps oxygen to leave the tissue, in other words. An increase in all factors, meaning an increase in the partial pressure of carbon dioxide, an increase in hydrogen ions, an increase in temperature, an increase in something that we call 2,3 BPG. Some books use DPG, so I'll just write diphosphol something, let's write, and then diphosphol something. But all these increases will cause a right shift, right? This causes a right shift from normal. This is known as the Bohr effect. The only thing that will decrease when you have an increase in partial pressure of carbon dioxide or an increase in hydrogen ions, we'll say in the blood, what goes down? It makes you more acidic, so the pH goes down. So you have to remember that the only thing that goes down is the pH. And that stands to reason. If carbon dioxide is up in the blood or increased in the blood, hydrogen is increased in the blood, that's going to make you acidic. And when you're acidic, you have a low pH, so the pH will be lower. Just remember that. Everything is up except for the pH, okay? So I'm just going to read this, that when the curve shifts to the right, like it did here, you have a decreased affinity of hemoglobin for oxygen. And this facilitates an unloading of oxygen to the tissues. An increase in all the factors except pH causes a shift of the curve to the right. Note that an increase in PCO2 decreases the affinity between hemoglobin and oxygen. This is called a right shift. A right shift of the oxygen hemoglobin dissociation curve when the PCO2 is increased is specifically known as the Bohr effect. PCO2 goes up, the curve shifts right, known as a Bohr effect. This is what you got to know. Lastly, I want to mention that, and I think I mentioned this before, but I'm just going to end with it, the respiratory control centers. The respiratory center in the brainstem controls our breathing, our inspiration and our expiration. This is known as the medullary rhythmicity area. Right? It's at the brainstem where the medulla is, medullary, and it controls the rhythm of our in and out, expiration, inspiration, our breathing. Now this concludes the lectures on respiratory. Our next lecture will begin with an entirely new chapter. It'll be after this respiratory exam, and we'll start with digestion. So after this, you'll start to look for digestion videos after the lymphatic and respiratory exam. I am going to write up there on the board what I explained about the red blood cell carrying carbon dioxide. 70% of it is carried by the bicarb. I'm going to just put it in writing so that you can understand what's going on with that drawing. And that'll be a frozen picture for you to just read. So that'll do it, and I'll get back with you with digestion.